Yes.
But there's like
live. Okay.
Yeah. Okay.
Exciting.
Yeah.
Never been live streamed before. I don't know if I like it.
Welcome to the big leagues. CSEE is definitely. All right. So now I'm going to post the link on the Twitter and the Facebook and everything. Is it as beautiful in Toronto as it is in Ottawa right now?
Yeah, it's amazing here. Been really happy with the weather.
Yeah.
Yeah.
It's crazy. Like 2 weeks ago, it was snowing here.
Yeah, here too.
Yeah. Like,
yeah. The weather's just been crazy. I mean, it's warmer. So I'm from Newfoundland, and so it's warmer there some days, just the past couple of weeks, than it has been here.
Okay.
Really unheard of usually.
Jeez.
Yeah. So has it been a lot of work organizing these talks to get them up and running on the live stream?
No, it is so much fun.
Yeah.
It's like I just get to like chill every Friday for 30 minutes, listen to some dope science, and like hype it up for the whole week. Like it's really brilliant.
Yeah, you're doing a great job on Twitter.
Thank you. Yeah.
Amazing. I've got so much publicity just from that Twitter going out every couple hours.
I'm happy. You deserve it for sure. All of you guys are just so freaking cool. It's awesome.
I enjoyed all the talks so far. They're so interesting. And it's fun to see all the different work that people are doing, but still within the field of ecology and evolution. Awesome.
Yeah, I love that about like these awards too. Like it's so, you get like the whole length of science or of like ecology and evolution.
I bet, yeah. It's fun to see all the different work that's being done in the field, especially with the sort of up and coming new researchers in the field, because
yeah. Do you have a title that I should introduce you with? A talk title?
A talk title?
Yeah.
You could say the effects of urbanization on species interactions. Okay. If you want. You could also just say whatever. I don't care.
Right. Yeah, we'll give it, one more minute and then we'll kick it off.
Yeah, it's fine. I want to make sure everyone hears my intro. It's going to be the best part of my talk. Is there any word on where CSEE will be next year, if it's held anywhere?
Oh, that's a good question. To be honest, I don't remember. We did have a spot for sure.
Okay.
I think it was in the West Coast.
Oh, that's nice.
But I forget exactly which campus, and it might not be there. It might have fallen through and been somewhere else.
That makes sense. I guess things are so uncertain anyways.
Yeah, that was even before kind of all this started. And now with kind of the COVID, I'm not sure how feasible it will be to have a big conference next year. But
yeah, I guess play it by ear, like everything else.
Yeah.
Life now.
Yeah. The theme of 2020 is definitely play it by ear. But on the subject of CSEE, we do have our election period that opens today.
Oh, I saw that email go out.
Yeah. There will be some tweets soon. Oh, and okay, it was on the West Coast. The meeting was supposed to be at UBC.
Oh, well, hopefully it can be there again at some point. If not next year, sometime soon.
Oh, and not just UBC. Could also be somewhere else. Maybe it's a joint thing.
Okay.
Let's see. All right. So I guess we're gonna kick it off now. Hello, everybody tuning in. Thanks for watching. This is, hopefully you realize if you're watching this, that this is the fourth CSEE Excellence in PhD Research Award talk. We had some excellent speakers over the last few weeks, and today we're really excited. I'm super excited to introduce our fourth speaker, Ruth Rivkin. She's going to be talking about the effects of urbanization on species interactions. And in a few minutes, I'll pass it over to her and let her take it over. But before I do that, I'm just going to announce that we have another talk next week for the CSEE PhD Award talk. And then the week after that, we have the Early Career Awards researcher talks on June 5th. So there's going to be some back-to-back talks from Dr. Denon Start and Dr. Rihanna Renninson on some really dope ecology and evolution research. So tune into those on June 5th if you've enjoyed the PhD Research Award talks over the last few weeks. And like usual in this talk, there's the live chat on the side. So if you're watching and you have a question for Ruth as she's talking, type in your question in the chat, and after the talk, I'll be here to moderate the questions and ask them to her and get you all an answer. So with that, I'm going to pass things over to Ruth. Thank you so much for being here, Ruth, and congratulations on the award. And take it away.
Okay, thank you so much. I'm just going to share my screen and then get started with my talk. Oh, it says that you've disabled screen sharing.
I am sorry. Yeah, that's the new Zoom default.
Yeah.
Not switch off.
Yeah, I like that they changed everything without telling anybody. Very convenient.
Yeah. All right. And now we'll pass it over to Ruth.
Okay. All right. So thank you so much to everybody for tuning into my talk. I'm disappointed that, we couldn't have this in person, but still really excited to have a chance to share some of my research with you. As Peter so kindly said, my name is Ruth. I'm a PhD candidate in the Department of Ecology and Evolutionary Biology. I work with Mark Johnson at the University of Toronto, Mississauga. And broadly, I study the effects of environmental change on species interactions. Today, I'm going to be telling you about how urbanization can shape the ecology and evolution of species interactions. So before I get started, I'm going to walk everyone through a brief outline of what this talk is going to look like. First, I'm going to cover some background about what exactly urbanization is and how I'll be using it throughout this talk. I'm going to give you the goals of my research project, and I'm going to take you through two studies looking at the effects of urbanization on plant-pollinator interactions and looking at the effects of urbanization on plant-herbivore interactions. And then I'm going to wrap this up by talking about how urban evolutionary ecology can be applied to the real world. First of all, what is urbanization? Well, the United Nations defines urbanization as a complex socioeconomic process that transforms the built environment. That's not super helpful for the sake of my talk. So what I'm going to define it as is any permanent human settlement that has humans shaping the environment that they live in. So we can have settlements in the form of cities, which are quite large and have, for example, 3 million people, like those found in Toronto. We have people living within towns, which are medium-sized and contain 100,000 people, for example. And we can have much smaller settlements, like villages and towns. But what's important about these three different types of urbanization is that they all create changes to the landscape. So urbanization is one of the fastest growing ecosystems in the world. And what this means is that we can have drastic alterations to the landscape. For instance, take this image of Toronto in 1984. I'm going to play through a video which shows the progression of the city development up until 2018. And I want you to keep an eye on one of the red outlines and just watch what happens to the green habitat found within them. So as we progress through the years, you can see that these green habitats become filled in and disconnected from the rest of the landscape until in 2018, they've either been completely lost, fragmented and isolated from the environment, or maintained in some smaller form than they were previously found in. So this, of course, can have drastic impacts on the ecology and evolution of species found within them. But we also have other types of environmental change that occur within cities. For instance, as I've just discussed, we can have habitat fragmentation and habitat loss, but we can also see elevated pollution levels within cities, both in the air and water, as well as in the soil and in the form of light and noise pollution. Lastly, we can also see increased temperatures, where cities are often several degrees warmer than the surrounding non-urban habitat. These environmental changes can have profound effects on the ecology and evolution of species. But let's break that down so it's a little more straightforward. First, environmental change through urbanization can affect the ecology by changing population sizes and densities of urban populations. We can see variation in dispersal patterns as humans move through the landscape and create barriers to dispersal through the formation of things like dams and roads. We can also see differences in species abundances, which can affect community assemblages, where we often see very different communities in urban sites than in non-urban sites. These environmental and ecological changes can affect the evolution of populations. For instance, we often find that the effects of genetic drift are stronger in urban populations because there's typically a lower amount of genetic diversity contained within those sites. We can also find evidence for elevated mutation rates in cities due to pollution. And we can find variation in gene flow, particularly in the presence of an urban barrier. Lastly, we can also see differences in natural selection. And this can happen when the urban environment selects for phenotypes and genotypes that provide an advantage in those changed environmental conditions found within cities. All of this evidence and more paints a very clear picture that the ecology and evolution of species is changing in cities. But what I'm particularly interested in is how this environmental change can affect species interactions. And that's what I'm going to be talking to you about. The core of my thesis centers around a single question: how does urbanization affect the ecology and evolution of species interactions? So we have two general scenarios for how we think this is going to play out. Let's take a straightforward plant-pollinator mutualism. Here, the pollinators and the plants are interacting in a reciprocally beneficial interaction. Under our first scenario, we might predict to see that urbanization decouples species interactions. This can happen through lower abundances and diversities of species that occurs due to urbanization, and this may result in fewer opportunities for the interaction. All of this can affect the fitness and evolution of traits and genes of those interacting species. Under our second scenario in this mutualism interaction, we might predict to see that urbanization intensifies species interactions. This can occur when urban habitat fragmentation reduces the amount of space that these species can occupy and causes more frequent interactions. And this again could alter the fitness and potentially lead to an evolution of traits and genes of those populations. So all of this is pretty straightforward when we lay it out in black and white on the screen. But it does get more messy when we think about how variable the urban environment can be. And so what I'm going to take you through is some of that complexity using two different sets of interactions. The first is an interaction between plants and pollinators, and this is a mutualism, which, as I've just said, is a beneficial relationship for two interacting species. I'm also going to take you through an interaction between plants and herbivores. This is something that we call an antagonism, and very frequently there's a benefit for one species at the detriment of the other species. So to begin with, the first study looked at how variation in pollinator-mediated plant reproduction varied across an urban gradient. This work was done in collaboration with Vanessa Nyan, who is an undergraduate researcher in the lab, as well as Art Weiss and Mark Johnson, who are both at U of T. Before I get into this section, I just want to go through briefly what I mean when I say an urbanization gradient. Many of you will be familiar with latitudinal gradients, where you go from a region of high latitude to a region of low latitude, and the environment changes along that gradient. We basically mean the same thing when we talk about urbanization gradients, where we're going from a region of high urbanization that is characterized by lots of built up areas and the presence of humans, to an area of lesser urbanization. And in this study, I'm going to be talking about that in the form of agricultural land. So in the plant pollinator literature, we often see two conflicting hypotheses. The first is what we've called the urban pollinator deficit hypothesis. Under this scenario, we predict that pollen dispersal and plant reproductive fitness are lower in cities relative to their surrounding non-urban habitats. And this can occur through habitat fragmentation that leads to a loss of an abundance and diversity of pollinators and reduced dispersal through the landscape. In contrast, we also see support for the urban pollinator enrichment hypothesis. Under this hypothesis, we predict that pollen dispersal and plant reproductive fitness are highest in the city compared to the non-urban surrounding habitat. And this could happen because urban lawns often contain nectar-rich resources in the form of dandelions and clover, and gardens in cities often contain quite a bit of floral resources for pollinators as well. And this can elevate visitation rates and increase plant fitness through those mechanisms. So we set out to test these two hypotheses. We asked two questions in this section. The first was, how does pollinator-mediated reproduction vary along an urban gradient? And the second, how does pollen dispersal influence plant reproduction along an urban gradient? We tested these questions using the lawns and backyards of volunteers who had kindly donated their property for use in this project. We did this project over 2 summers, and we repeated the experiment multiple times in each summer. At each of these sites, we measured various measures of urbanization. The one I'm going to be telling you about today is the amount of impervious surface in an area. What I mean by impervious surface is essentially any surface that isn't permeable to water and doesn't allow plants to grow through it. You can see in the pop out here, I've highlighted all of the surfaces that we've deemed to be impermeable. And these are, and we did this within 100 meters squared radius of the site. We measured this at every single site. And we also measured distance from the city center in downtown Toronto. We measured the number of buildings found within a 100-meter radius and the canopy cover. These variables were all highly correlated, so I'm only going to tell you about impervious surfaces, but happy to answer other questions about the other variables at the end. We set out at each site patches of rapid cycling brassica rapa. For those of you who don't know rapid cycling brassica rapa, it's an excellent system to do field experiments with, because it goes from germination to flowering in about 14 days and then produces its seed in 60 days. So it's ideal if you're doing short-term experiments in a short-term summer climate. It's visited by a whole bunch of different pollinators. It's pollinated by generalist species typically. And we, for example, we observed bees, bumblebees, hoverflies, wasps, and butterflies pollinating these plants. We used 2 lines of rapid cycling brassica rapa to test our questions. The first was a standard hermaphroditic line. This was our measure of plant reproduction. The hermaphrodite plants are self-incompatible, so in order for them to set seed, they had to receive pollen from another hermaphrodite within the patch. We also used a male sterile line, and this was our measure of pollen dispersal. The male sterile plants can't produce pollen, and so in order for them to set seed, they had to receive pollen from a hermaphrodite within the hermaphroditic patch. At each of the sites, we created 2 patches. The first was our hermaphrodite patch. We had 5 plants here, and as I've just said, they needed to receive pollen from one another in order for them to set seed. 5 meters away, we created a second patch of our male sterile plants. These plants can't produce their own pollen, so in order for them to set seed, a pollinator first had to visit the hermaphrodite patch, then disperse 5 meters to the male sterile patch. And in this way, the difference in seed set between the male sterile and the hermaphrodite patches was our measure of pollen dispersal. So hopefully that's all clear to you. I'll just remind you that we are testing how reproduction and pollen dispersal vary along an urban gradient. First, we found that plants opened fewer flowers in the city. I'm going to show you a number of graphs that look like this, so I'm just going to take a minute to walk you through this. On the y-axis, we have the average number of flowers produced per plant per site. And on the x-axis, we have the impervious surface in a site. Here, all you really need to worry about is that higher values of impervious surface corresponded to more urban sites. So we found that flowers opened fewer flowers, we found that plants open fewer flowers in urban sites and more flowers in rural sites. And this result was consistent with our urban pollinator deficit hypothesis. In contrast, when we looked at seed set, things got a lot more complicated. Here on the y-axis, we now have the number of seeds per plant, and on the x-axis, we have the impervious surface again. The colors are broken up into month, and in June, we found results that were similar to those for flower number, where plants set fewer seed in urban sites compared to rural sites. But as we move through the month, this trend changed. In August, the line flattened out, and by September, it had completely reversed itself, where we saw more seeds being set in urban sites than in non-urban sites. This result is consistent with both the urban pollinator enrichment hypothesis and the urban pollinator deficit hypothesis. Lastly, we found that pollen dispersal decreased with urbanization. Again, on the y-axis, we have the number of seeds set per plant, and on the x-axis, we have the impervious surface. And I'll just remind you that our estimate of pollen dispersal was the difference in seeds set by the male sterile plants in pink here and the hermaphroditic plants in orange. So we found that this difference in seed set was greatest in urban sites, which suggests that pollen dispersal is lower in urban areas. This result is again consistent with the urban pollinator deficit hypothesis. So in summary, we found support for both hypotheses. We found that our results for flower number, early summer season seed set, and pollen dispersal supported the urban pollinator deficit hypothesis. And this could be due to elevated pollution at urban sites, as well as habitat fragmentation in those areas. However, we also found support for the urban pollinator enrichment hypothesis in our results for seed set in the later summer. This could be due to seasonal turnover in pollinators and perhaps additional floral resources being provided by ornamental gardens and lawns in cities. But what I really want you to take away from this is that urban habitats are complex, and we often need to extend our studies over multiple years and across a season in order to really understand how things are responding within them. So that brings me to my second study, and I'm just going to take a quick drink of water. So here in this study, we looked at the effects of urbanization on plants and herbivores. So in this study, we looked at how urbanization affected interactions between Darwin's finches and Tribulus cystoides on the Galapagos Islands. This work was done in collaboration with Jaime Chavez, Mark Johnson, and Reagan Johnson. They were the ones who did the field work here. And for any grad students who have had their projects put on hold because of the COVID pandemic, just wanted to say that I feel your pain. I'm right there with you. And sometimes these interim projects can turn up some really interesting results. So compared to mutualisms, we know a lot less about what's going on with antagonistic interactions in urban areas. We know that trophic interactions should be susceptible to urbanization. And I'm just going to walk you through an example of what I think might happen. So let's say we have a 3-level trophic web where we have the top level consumer of birds, we have an insect herbivore, and those feed on the bottom level producers, which are plants. If we have a disturbance that knocks out interactions and knocks out the presence of birds in the landscape, we might expect to see an increase in the abundance of our insect herbivores. This in turn can cascade into altered selection pressure and potentially lead to an evolutionary response on the plant populations that those herbivores feed on. But again, we know very little about what's going on. And so we set out to test some of these predictions using populations of Darwin's finches and Tribulus cystoides. So the Galapagos Islands are a small archipelago off the coast of Ecuador. They consist of a bunch of islands that form up this archipelago, and they're inhabited by a small number of people. The cities that are found there have been formed relatively recently, and they have relatively small human population sizes. But we wanted to test how these interactions changed on the islands for 2 reasons. First, islands should be particularly susceptible to disturbances, partly because they have these unique ecosystems, and partly because there's really no escape for these species. They can't easily disperse out of those sites. Second, because of the relatively recent colonization history of the Galapagos Islands, they allow us to test how urbanization shapes species interactions when it's at the very beginning stages of occurring. So these are relatively new cities, and they offer us a chance to understand how these species are responding to the beginning levels of disturbance. So we looked at interactions on 3 islands, and we chose these islands because that's where the large-beaked finches and the populations of Tribulus cystoides are found together. These islands differed in population size. Santa Cruz was the largest with a population of 12,000 people. Isabella had a population of 2,000, and Floriana was tiny with only 145 people living in the town there. Many of you will be familiar with the Darwin's finches, but for those of you who aren't, this is a species that has undergone a very rapid radiation, and partly due to beak size and diet preferences. We were interested in 2 species, the large ground finch and the medium ground finch. Both these birds have large beaks that are capable of cracking open the hardest and the biggest seeds on the islands. And they are a primary predator of Tribulus cystoides. This plant is also known as the puncture vine, which, as you can see from the image at the top of the screen, they have these really big spines that act both as a dispersal mechanism and as a mechanism of defense against predation. But they're not safe from large beak finches. The birds will come in and they'll grab a fruit from the ground, and they'll crack it open using a shearing motion of their beaks. This produces a lot of damage on the fruits, and often they will have they'll be able to crack off the ventral surface and access the seeds within them. But there's a lot of variation in the level of damage that these fruits take. We wanted to test interactions between these 2 species for 2 reasons. First, both the Darwin's finches and Tribulus cystoides have been shown to be influenced by humans. Humans are the primary dispersers of Tribulus fruits. And Darwin's finches have been shown to have different diet preferences in urban areas where they prefer human foods like chips to their natural food resources. Second, we know that finches impose selection on Tribulus fruits where they are selecting for both smaller and harder fruits. So we know that there's a lot going on here, and we think that urbanization may be shaping some of these interactions between the species. Here we asked 2 questions. Our first question, does seed predation by finches differ between urban and non-urban populations of Tribulus cystoides? Our second question, does urbanization alter selection imposed by predation on fruit morphology? And I know I said 2 questions, we actually had 3 questions. Does urbanization affect the community structure of the Galapagos finch populations? I don't really have time to get into this 3rd question, but again, I'd be happy to talk about it in the question period. We tested these questions using a combination of natural population surveys and field experiments. For the natural population surveys, we went out to natural, urban, and non-urban populations pictured here of Tribulus cystoides. We visited a total of 85 populations across the 3 islands and collected between 20 and 40 fruits per island. On each of the fruits we collected, we counted the number of seeds that had been eaten from them, and we measured various aspects of their morphology. So as you can see from this image, the fruits are pretty difficult to describe in a single or just a couple traits. So what we did was we took a lot of different measurements and we distilled those down into 2 traits using a principal component analysis. We measured the traits for size and for defense, and these were the traits that we used in our model. So these are composite traits, not a single, and they consist of multiple measurements that have been distilled down. For our field experiment, we selected urban and non-urban sites across our 3 islands. At each of the sites, we placed out trays pictured up top there and left them out there for 6 weeks. We selected 60 populations per island, or 60 populations total across the 3 islands, and we set out 20 fruits per population. Before we set out the fruits, we measured their morphology and calculated those same two traits that we calculated in the natural population survey, one for size and one for defense. We marked the fruits so we could track which fruits were removed by finches during the experiment, and we clipped the spines from half of the fruits. So this is a natural state that these fruits are found in. Often you find them without any kind of defense. But what we were interested in was understanding how the artificial removal of defense affects their likelihood of being eaten, and also how that might change with urbanization. So we set the trays out and left them for 6 weeks. And you can see in this bottom image, the birds will come in, they'll land on a tray, and they'll carry off fruit to eat at another location. And so this was our measure of predation. So again, I'll just remind you briefly, we are looking at how seed predation and selection on fruit morphology differ between urban and non-urban sites. And I'm going to take you through the results from the natural population survey first, followed by the results from the field experiment. So first, we found that seed predation varied across the sites, but this effect depended on island. On the y-axis, we have the number of seeds eaten per fruit, and on the x-axis, we have our islands. The islands are ordered by human population size, where Floriana has the smallest population size and Santa Cruz has the largest. Urban and non-urban populations are shown in blue and in red. We found on Isabella that there is increased predation rate in urban populations, where fruits had more seeds eaten per fruit than in non-urban populations. We found the opposite trend in Floriana and Santa Cruz, where predation rate was higher in non-urban populations than in urban populations. We also found that there's an effect of fruit size on predation rate, and this effect was intensified in urban populations. On the y-axis, again, we have the number of seeds eaten per fruit, and on our x-axis, we have size. You don't need to worry too much about the numbers. Just keep in mind that negative values represent smaller fruits and positive values represent larger fruits. Overall, we found that there was an effect of fruit size on the number of seeds that had been eaten per fruit, where smaller fruits had more seeds eaten from them. However, there's also an effect of urbanization. Urban populations, shown by the blue line here, experienced more predation than did non-urban populations, and the effect of this predation was stronger in those sites. What this suggests to us is that by consuming more seeds from small fruits, the birds are imposing selection against small fruit sizes and potentially triggering the evolution of larger fruits. Moving on to the experimental results, we found a direct effect of urbanization on the probability of fruit removal. Probability of fruit removal is on the y-axis and site on our x-axis. So we found that fruit removal was higher in urban populations than in non-urban populations, and this effect also differed among islands. But unlike in the natural population survey, there wasn't an interaction between island and site. We also found that there was an effect of removing the spines on the probability of fruit removal from the site. So we found here that clipping the spines increased the predation rate experienced by those fruits. However, we didn't find an effect of urbanization. So there was no effect. So this effect of removing the spines from the fruits didn't change whether or not a fruit was in an urban or non-urban site. And this is very interesting because we actually did find an effect of natural defense on fruit removal in urban sites. So here on the y-axis, again, we have our probability of fruit removal. And on the x-axis, we have our defense trait. Again, don't worry too much about the numbers on the axis. Just keep in mind that negative values represent undefended fruits and positive values represent defended fruits. Here again, our sites are broken up into urban and non-urban populations. And what we found is that predation was higher on well-defended fruits in urban populations and lower on well-defended fruits in non-urban populations. Similarly, predation was lowest on undefended fruits in urban populations and highest in non-urban populations. Because urban populations experience more fruit removal than because defended fruits in urban populations experienced more fruit removal than did undefended fruits. What this suggests to us is that there may be evolution for undefended fruits in urban populations. And we don't predict this in non-urban populations because we see the reverse trend occurring. So in summary, we found that urbanization impacts predation and triggers selection on fruit morphology. Based on these results, we predict that urban tribulus fruits may evolve to be both larger and less well defended. And we think this may have something to do with the finch community. So we found differences in the diversity and abundance of the finch community, both across sites and among the islands. And we also found that there is different community structure among those sites. And so this suggests that the presence or absence of certain types of finch species may be contributing to evolution within the urban sites. So I'd like to wrap up my talk with a brief discussion about the applied aspects of urban evolutionary ecology. Urban evolutionary ecology can help inform projects on community engagement and education. For instance, we could set out educational signage in urban field sites to help explain to the public what exactly we're trying to look at. We can also do easily accessible community engagement projects, such as what I did with my brassica study. We can use urban evolutionary ecology to help inform conservation practices. For instance, we could plant sufficiently diverse and beneficial gardens that help promote the abundance and diversity of native pollinators. It can help with urban design, such as designing parks that allow for connectivity among populations and promote the dispersal and migration of alleles across the environment. And it can help inform pest control practices by helping to target and promote the most efficient ways to remove pests from the urban landscape. So with that, I'd like to thank everybody, everyone in the EVO Eco Lab, past and present, all my collaborators, and as well the private landowners who are involved in the brassica study. I'd also like to thank my funding sources. And I'll just finish up by saying, I'm thinking about doing a postdoc in the fall of 2021. So if you're looking for a postdoc and think my work is interesting, please feel free to reach out. Thank you.
Awesome. Thank you so much, Ruth. That was an amazing talk. I hope your shout out for a postdoc works. We'll definitely add it into the Twitter promo after. But fantastic job, and thank you so much. I'm curious, just offhand, did you get to do any field work for this in Galapagos Islands?
No, sadly, I didn't. This really was a COVID project where the data set was there, and Mark gave it to me to analyze and to play around with. I wish I could have done the field work. It would have been amazing, but sadly, no.
Well, great job nonetheless. So a few questions, a lot of questions rolling in, a lot of congratulations as well. First question, regarding the first part of your talk. What was the buffer diameter of 100 meters squared for impervious, or why was the buffer diameter 100 meters squared for impervious surfaces?
So for a lot of the pollinators we were working with, the generalist pollinators, their dispersal radius is about 100 meters. It can be larger for certain pollinators and much smaller. So we kind of averaged that. And it was easy. We had a lot of sites and a lot of sites within a relatively small area in downtown Toronto. So we needed to find a measurement that both allowed for pollinator movement and also didn't overlap among sites.
Awesome, thank you. Another question. The brassica results are really cool. I also agree with that. I think they're really cool. Do you know if the farmers grow canola or similar plants nearby, and whether these plants could have been pollen donors for the experimental arrays?
Yeah, so I think there is some canola, although I don't think that there was a lot. But there certainly wouldn't be canola within the city, so it wouldn't necessarily explain why we saw that trend in seed set occurring and changing over the season towards more urban areas. Also, we tried to select sites that weren't too close to any like really active farming areas. So hopefully we were able to avoid that.
Did you know, did you monitor the pollinators that visited the plants at all?
We did, yeah, actually. So that was Vanessa's section, was really working hard on the pollinator observations. We looked at a subset of sites and we saw trends towards lower abundances and diversities in the city, but higher abundances in the later summer. But we didn't really have the power to detect any kind of significant trends. That being said, there's been a lot of work looking at urban pollinators, and the results are all over the map. It really depends on which species you're looking at and when you're looking at it, where you're looking at it. Yeah, I could write a whole review just on that. I'm pretty sure people have done that before.
Yeah. We'll look forward to it though. Another question regarding the seasonal turnover of pollinators. Did you record, oh, this is literally, sorry, the question we just asked. Did you record the presence of different pollinators from different seasons? And I guess, do you know what species tended to drive the seed set observations?
We don't have direct correlations for that. And to be honest, we don't know which species were really driving it. I've done some other projects with some people in the lab where we find that bumblebee abundances are much higher in cities. So it's possible that because they're doing so well in cities, that they're the primary pollinators. That being said, just sitting out there, I had one in my backyard, sitting out there watching it, there's a lot of syrphid flies, a lot of different things coming and going. So it is fairly difficult. But to answer the first question, we did track it across the season, yeah.
Within the highly urban environments, what factors do you think contributed to the observed variation in the seed set?
That's a great question. So there could be a lot of different things going on. First, it could be that habitat fragmentation is triggering less movement between flowers in urban sites. So for instance, we often see when patches are further apart, they will have, the pollinators will kind of maximize their time spent in any one patch and they'll move less between patches. We didn't have any kind of habitat fragmentation occurring between our patches, but it could explain differences among the sites. Across the season, I do think it's seasonal turnover in who's around in the summer in terms of the pollinators. The gardens are blooming all the way through September, and often we see people kind of plant May 2-4, so then everything kind of blooms later in the summer. So there's just more resources, and it's possible that we see more pollinators or different pollinators that are better at pollinating our plants.
Thanks. Another question for the tribulus experiments. For the antagonistic interactions, do you think that predators may evolve faster than the resources in urban environments?
That's a good question. I don't necessarily know. I think it depends on a lot of interacting factors. For instance, predators are often, if we're talking about the top trophic level, there's no top-down effects on them. So you might expect they might almost be relieved from disturbance pressures unless it just knocks them out or has some kind of direct effect on them. Whereas the things that are getting eaten, or the consumers, I guess, or the prey, that's the word I'm looking for, the prey, those can be influenced by top-down effects through disturbance on the predators, but they can also be influenced by bottom-up effects through effects on the food that they consume, if we're talking about like an herbivore or something. For the stuff that I've seen that's out there, we see a lot of people tend to look more at sort of the mid-level individuals, although I have seen some coyote studies coming out, which show that there are strong effects of urbanization occurring on those populations.
Cool. I'm curious whether you think there would be always a linear relationship in urbanization, or I was thinking kind of, sorry, I was thinking specifically, especially these like really kind of naive like urban sites, like in the Galapagos, when you introduce like cats or rats or other things that might prey on these predators. Do you think that has an impact? And do you think it maybe did in your study as well?
I think it probably did. So what's interesting is that we saw different effects depending on what island we're looking at. So on Floriana, which is our smallest, and I don't know if it's the most recent, but it's definitely the tiniest in terms of size, human population size, we saw that there were equal abundances and diversity in the urban and non-urban sites. Whereas on our larger islands, we saw that diversity and abundance were lower in urban sites than on non-urban sites. So to me, that would suggest probably there are effects of larger cities that we're just not capturing in Floriana because it's too small, which is also interesting because there could be, ways that we can try to modify our cities to reflect small cities within small towns within a large city. But so yeah, I do think that there is an effect of those introduced human cohabitors.
Someone is interested to know more about the community structure analysis.
Yeah, so this was fun because this one I had to really dig into the stats literature and do things I've never done before. So we ran an RDA analysis, a redundancy analysis, where we took a community matrix of our species abundances, and then we took a matrix of our environmental conditions, which in this case was urban or non-urban, our 3 different islands, and then the populations to just control for differences among populations within a single island. And we regressed those 2 things. And so what we found was that the communities were more constrained in urban sites. So we found, and interestingly, we found that small finches really fall out as an outlier in urban sites where they don't fall out at all in non-urban sites. I don't really know what's going on there. They don't consume tribulus, so it's probably something to do with the food resources in urban sites. Perhaps if people have bird feeders out, they may be capitalizing on that. So that could be something that's going on. And then we do see effects of our islands as well. So again, Floriana falls out as much more similar between our 2 different environments, whereas the other 2 islands there's a lot more constraining happening within those communities.
Okay.
Yeah.
And what types of interactions, I guess, did you include? Was it just similar to before?
In the RDA?
Yeah.
Yeah, so we included the interaction between site and island. And again, we saw a significant interaction. It gets really hard to understand what's going on among islands within the sites, but I think it's Floriana that's acting much more like a natural population than an urban population. I think that's what's going on, if that helps.
Thanks. There seems to be some evidence for divergent natural selection between urban and rural environments in both studies. Can you speak to how this environmental divide could lead to local adaptation? Yeah,
so I do first want to say that our results from the Nebraska study reflect phenotypic plasticity because they didn't come from those environments and we only put them out for a short period during their life cycle. That being said, the studies that have looked at local adaptation find some really interesting results. For instance, I can think of at least 2 that find that while there is local adaptation happening to urban environments, we actually see that there is maladaptation happening in some of the traits that people looked at where the phenotypes are changing in a way that doesn't really benefit those populations in urban sites. We also have some limited evidence for increased plasticity in urban sites, although I think more studies need to be done to really confirm how that's working. And we see a lot more support for the urban heat island effect than any other kind of change in the environment in urban areas. I think maybe because temperature just has such a drastic effect on species in general, but we do see that there is adaptation happening to elevated urban temperatures and quite rapidly too, and across different taxa, which is one of the most interesting things, I think, because we see this happening across multiple taxa and systems and cities. So it's clearly a case of parallel evolution if we're talking about how multiple systems respond to the same pressure. And I think that's very fascinating.
Definitely, yeah. One more question. Did you see any trends in bees versus syrphids in urban versus rural?
So not very directly in this Nebraska study, but I have been collaborating on another study also across the Greater Toronto area where we saw basically conflicting trends in bumblebees, where we saw higher abundances in cities and lower outside of cities, and higher abundances of honeybees outside the city, lower in the city, which kind of makes sense if you think that honeybees are really pollinating agricultural systems. But when we looked at syrphid flies, we found no trend. So we basically see cross lines and then just a flat line. I don't really know how to interpret that quite yet. We're still working on that one, but I hope that's as interesting to other people as it is to me.
Yeah, definitely. That's really, really weird. Yeah.
Yeah.
One final question, just to round out the period. This person is interested in your seed set data in urban sets versus rural sets. Do you think that there may be variation in phenology between rural and urban areas, driving a late season increase in seed set in the urban environment, just due to microclimatic differences?
I think that probably does exist in natural populations. I think some people out at Becky Irwin's lab are working on that. In our study, because we basically took common garden plants that came from mostly the same source, put them out in urban environments, brought them back in, it's not necessarily going to be due to variation among the plants themselves as part of like their genetics. It's much more likely to be phenotypic plasticity. For the flowering number, we have some evidence to suggest that plants are damaged by volatiles released by car emissions. So that might be why we saw fewer flowers opening in the urban environment. And then for seed set, I think it has to do with how well they're getting pollinated in terms of pollen limitation as well.
Okay. Awesome. Thank you so much. Terrific job, terrific presentation.
Thank you.
Excellent job answering all these questions. Very happy we could do this online.
Yeah, well, thank you so
everybody.
much for organizing this. This has been really great. It's nice to have a chance to still share my results.
Yeah, we're happy to hear it. Yeah. Thank you everybody for tuning in. Thank you for your excellent questions, for your virtual applause, of which there was a lot. Thank you. And just a reminder to everybody, our 5th and final PhD award talk is next week. And we're also super excited to see that one. And then the week after that, on June 5th, there will be the 2 early career award talks that will be going on back to back on Friday, June 5th. Both from one from UC Davis, one from UC San Diego, giving some excellent talks on evolutionary ecology and ecology. And it will be no doubt also a spectacular series. So please stay tuned for that. Thank you all for tuning in, and hopefully we'll see you all next week.